Hello, everyone. We are still expecting several more guests, but I just want to let you know that we'll wait a minute, but we do have some housekeeping and announcements, and I think I'm going to start in with those. And then Robbie wants to say a few words, and then we'll introduce our guest. So as we did just say, we are recording this session. So, careful what you say, just know that it will be available for others to view later, probably in a week or so. During the presentation, please keep your microphone muted until you are recognized. And please type your questions in the chat box. We will be monitoring those so you won't be missed. We'll get to all of the questions, we should be able to. And then Q&A will be held until at the end of the presentation, but there will be ample time for a good Q&A session. And then as far as upcoming, well, let me just first say that our 2022 Hertz application is open, now available on the website, and it is available until October 29. Please share the application with anyone you think is interested or qualified. And if you did not receive the application announcement, which went out last week, please be sure to let us know and we'll get that to you. We would love for you to help promote that to others. And then our next innovation hour will be on October 13th with Hertz Fellow Leonid Krugliak, who will present on SwabSec, which is a solution for large-scale COVID-19 testing and other things. So please put that on your calendars and we hope to see you then. So at this point, Robbie, I'll pass it over to you.
Thanks very much, Carla, and a good afternoon to everyone. I know some folks are still joining us. I can see the counter continuing to move up. I just wanted to add my thanks and my welcome to Carla. It's nice to have all of us be able to get together like this and share great science and technology breakthroughs like Chris is going to be sharing with us today. But we all know that being together in person is really the most powerful, although this may be the most convenient. But I am delighted to say we are marching on at the foundation and continuing our quest to be able to bring Hertz Fellows together in ways that are safe and comfortable for everyone. We are in the midst of planning for the summer workshop for next summer. That's July 21 to 24. So mark your calendars, July 21 to 24 in Boston. And we're going to be at the Seaport Hotel there. God willing, pandemic willing, climate willing, we will be there and in person, and a really terrific program is beginning to come together already for you. So I hope a lot of you will be able to plan your summer vacations around that and join us. And I did want to emphasize Carla's mention on the fellowship application. You all as Hertz Fellows know better than anyone the talent and drive and creativity that we are looking for to award our fellowships. And so if you do know that special young person, or if you do know people who might be working with such folks, I do hope that you will pass the application link along to them, or just direct them to the website, and you can easily see there's a link that sits on the front page of the website as well. But I am going to tell you, this is going to be an even tougher season for our interviewers. So those of you who are interviewers and joining us today, strap yourselves in. We already have over 900 students that have started applications for this year. Now, we all know that some students start and for a variety of reasons end up not completing those applications. But given the data that we have, this is the highest number of applications that we have already had started at this point in the application season. And it's with just a peek into some of the information that's already in there. This is going to be a really strong group. So we're going to have our We have our fundraising challenges set out for us to raise even more money so we can award more fellowships to deserving students, and the interviewers certainly have their work cut out for them to make those choices. So I want to thank you all for joining us. I hope you'll continue to join in Innovation Hours, and I look forward to seeing you as we begin to open up in-person activities around the country. So thank you very much.
Okay, and at this time, it is my pleasure to introduce our guest speaker today. I'll read a bio to introduce him and then hand it off. Hertz Fellow Chris Own is the founder of Voxa, where he creates instruments that catalyze new technologies and promote improvement of the human experience. He is an expert in analytical design and development with specialties in advanced imaging systems and science instruments, ranging from atomic resolution structural imaging to portable charged particle-based analyzers. Trained at Northwestern University, where he received both his Bachelor of Science and PhD in Material Science and Materials Engineering, his career spans thin film research, electron crystallography, materials characterization, biological imaging and informatics, and spaceflight instrumentation. Chris has published in broad areas with over 50 publications, book chapters, and patents. He has launched 3 companies and worked with startups to commercialize novel charged particle-based automated nanoscale imaging and characterization technologies. His current teams have helped researchers produce some of the largest anatomical data sets ever created and has sent the first electron microscope to space. We're delighted to have you kick off our fall season of Innovation Hours, Chris. Thank you for being here and a warm welcome to you.
Thank you very much, Carla, and to the Hertz Foundation for the invitation. It's a great honor to have the chance to share our work at Voxa with the Hertz community today on production and democratization of some of the largest scientific data sets in the world. I'm CEO and founder of Voxa, and we're in the business of measurement and analysis. So to set the tone, I'd like to open the session with a quote by the Venerable Lord Kelvin on the topic of measurement. He said in 1883, I often say that when you can measure what you are speaking about and express it in numbers, you know something about it. But when you cannot measure it, when you cannot express it in numbers, your knowledge is of a meager and unsatisfactory kind. It may be the beginning of knowledge, but you have scarcely in your thoughts advanced to the state of science with a capital S, whatever the matter may be. And so in the way that only a late 1800s Lord could say, in modern day times, you might say you get what you measure. In other words, it's of great importance to accurately assess things and putting numbers to phenomena in order to understand them and use them properly. So in line with Lord Kelvin's sentiments, a little bit about our company Voxa and myself. Our mission is to make precision measurements and imaging more widely available and push it into frontiers where it's never gone before. And currently in the form of novel commercial instruments with focus on imaging tools at the moment. Voxa is still a fairly young and small company. Our core wheelhouse is in cutting edge industry instrumentation and automation, and we've had some pretty good firsts. We built the world's first and only portable scanning electron microscope. It's about the size of a coffee maker, and it runs on an iPad. We've also built the first fully automated and scalable transmission electron microscope pipeline, which would be the main topic for today. And further, we placed the first combination electron microscope and X-ray spectrometer in space. To echo the line in the intro from Carla, I've started a few profitable businesses. And the first was in my second year of grad school while on the Hertz Fellowship, and I was building hi-fi digital audio electronics. And I joke with Carla that I was so worried I would lose my fellowship that I kept it a secret until long after I received my degree and felt like I'd accomplished something in my professional life. But regardless, after that, I was hooked on business and knew my calling was to work in small companies on big ideas. And so following graduate school, I built advanced retrofits for electron microscopes at a company called Beam Devices that I founded. And then afterward, I decided to be an individual contributor in a couple startup companies. First, it was a highly technical electron microscope company in Seattle, and the other was a Silicon Valley company. It was a fast-paced genomics startup funded by high-profile Bay Area investors. My background and training is in applied physics and engineered systems, and so it enabled me to start Voxa And so Voxa offers both high and low end resolution imaging tools. We call them Mochi and Blade, and we also cover portable as well as big data applications, and also provide services terrestrially and on the International Space Station. Voxa's goal is to help people scan effectively the infinite nanosurfaces of the world and make that data more accessible. So I just wanted to take a quick moment. This isn't the main topic for today, but one example of how our technologies are at the frontiers is providing access in outer space. So just want to take the quick opportunity to let you all know that we welcome you to propose research in space, making use of this unique national laboratory facility on the only microgravity research platform in the solar system. So it's going to be booted up pretty soon, and we're still in process of making it available, but it's we're pretty excited about the prospects of helping people do characterization in microgravity. So now let's return our focus back to Earth. So one of the other great frontiers of the nanoscale is the human brain. And it's no secret that we know very little about the brain and its inner workings. We know that the brain is a sophisticated pattern matching engine operating at about 1 Hertz. It's able to perform fabulously complicated computations running on just 20 watts of power in humans. We also know that most computation in the brain occurs by virtue of its interconnections via multiple hierarchies, ranging from electrical pulses to transfer of complex proteins and sugars. So a handful of natural computational systems are currently understood. The olfactory system being one of them is a beautiful example of textbook compressed sensing circuits. And also some retinal circuits are tuned to have simple sensitivity to first and second derivatives of contrast, which provide an evolutionarily programmed edge and movement sensitivity. But despite this knowledge as a whole, simply stated, we know very, very little about how it all works, even less about how it breaks. So doing a quick internet search, it's easy to see this lack of knowledge is broadly acknowledged. So here are just a few quotes from a quick search. Neurobiologist Lu Chen from Stanford says, we know very little about the brain. We know about connections, but we don't know how information is processed. Understanding exactly how the neural pathways function could lead to improved treatments for depression, genetic disease, and many other conditions. Her colleague, Nobel laureate Tom Sudoff, who's also at Stanford, says, medicine is a craft. It's empirical, but we don't know how to treat problems if we don't understand the disease and underlying biology. And further, New York Times reports to a more general audience, so many large and small questions remain unanswered. How is information encoded and transferred from cell to cell or from network to network of cells? Science found a genetic code, but there is no brain-wide neural code. No electrical or chemical alphabet exists that can be recombined to say red or fear or wink or run. And no one knows whether information is encoded differently in various parts of the brain. So a chronic undercurrent in our day-to-day lives is the common disorder of our neurological systems. Illnesses that societally are of epidemic proportions, and so in a post-scarcity and more information-filled world, we find that mental health issues are becoming increasingly prevalent and impact almost every member of our society yearly. And the numbers are astounding. It's 1 in 4 adults per year. And many adults suffer more than one disorder. So there's lots of comorbidities. And depression is just widespread. And despite its prevalence, mitigations are actually sorely lacking. So a case in point is central nervous system or CNS drugs. In a survey from 2010, CNS drugs were found to be the most expensive, about $1 billion each to develop, least efficacious, and also take the longest, which is about 10 years of all major classes of drugs to develop. So therefore, it's crucially important that we increase our rate of gain of knowledge of neurological systems, of which we currently have very limited understanding. So here are a few examples of current tools that are useful to aid in our understanding of neural circuits. Available tools include others like CAT scans, MRIs, and genetic sequencing, microarrays, proteomes, that sort of stuff. But I'll just focus on these because they're more easily measurable. Some of these tools directly measure activity and others measure indirectly, but some are also easier to use than others, and some have high resolution and others don't. In the second half of the last century, fMRI and electrical stimulation were very popular. And concurrently with the rise of cheap computation and improved imaging tools, circuit theory and AI algorithms have also made great strides, as everybody should probably be pretty well aware. So our focus for today is on imaging as a key measurement modality. As a trained material scientist, I was surprised that biology researchers had their hands full with reproducibility and confounding issues due to high variability of biological processes in naturally occurring systems. Especially in the solid state for material science, the general expectation is for consistency of systems across samples, and it made it easier to deliver consistent measurements. But in biological sciences, this just isn't true. There's so much variability. And looking at it for myself, I felt this was a place where increased sensitivity and automation, which were things that I had experience with, could provide significant gains to us. And extending on this thought, a crucial way to aid in consistency and in our understanding of the brain is in making maps. So looking back to history for some cues, we can ask, why was the development of cartography so important during the age of exploration? In the 15th century, the countries of Europe sent people all over the world exploring. Map makers became important, and maps made new information easily visible. So even people who never went anywhere could learn and be inspired. And maps also helped explorers go back to places they had found by accident. And so by mapping territories, explorers massively increased information flow and the wealth of opportunities that came with it. And faster transportation and better timekeeping technologies vastly improved exploration abilities and the accuracy of maps. So by analogy, better quality maps of tissue systems has similar potential to exponentially increased discovery in these types of systems. And I'm a very firm believer in this, of basically making the data as free as possible, and lots of it. So a primary theme here is to leverage newly available scientific tools, which offer multi-order of magnitude performance gains and rapidly increase the scope of possible learnings about neural circuits and systems. And that gives potential for elucidating huge variability within natural systems, discovering novel patterns and hierarchies, measuring efficacy of new therapies, and also looking at the computational side, verifying existing computational and theoretical models, and finally also informing new models of organization and computing, which is in part how this project was funded. So emphasizing the visual more, it's important to remember that modern neuroscience sprang from direct visual study of tissue structures. Many of you may know about Ramon E. Cajal already. And the first time I heard of him and his work, I was just completely floored. He's considered the father of modern neuroscience and dreamed of being an artist in his youth. And his father would have nothing of it and discouraged him from art. And being the dutiful son, Cajal took up a scientific career channeling his artistic gifts into his science. So he used the methods of Golgi. He was a contemporary of Golgi, and Golgi had pioneered stains. And he stained unmyelinated neurons, identifying a huge diversity of cell types. And so in 1880, what Cajal was doing was he was taking pictures. He didn't have digital cameras or film. He just simply drew what he saw with meticulous precision by looking through the ocular on the microscope. As a result, many of Cajal's insights and conclusions on structure and function are still valid to this day. So moving ahead half a century, much more powerful imaging tools became available. The community started asking new questions and going smaller and smaller. So in 1959, Feynman gave a seminal talk that many of us know, kicked off the field of nanotechnology. It was very inspiring to me when I was a student. And in it, he has sections specifically devoted to electron microscopy. So he asked, what are the most central and fundamental problems of biology today? They're questions like, what is the sequence of bases in the DNA? Where do the proteins sit? Where do the amino acids go in? It's very easy to answer many of these fundamental biological questions. You just look at the thing. Make the electron microscope 100 times more powerful, and many problems of biology would be made very much easier. And so what he was saying was that even the most advanced microscopes of the time still needed to be better. And importantly, hidden between the lines there is, as the length scale decreases, the detail of maps increases. So it's important to remember that by increasing the resolution, the information density increases by the square with the area and by the cube with volume, which makes the amount of data just blow up incredibly quickly. And this leads to the question of what if imaging was made better? even in this day and age, and much better. And we're talking multi-orders of magnitude better. And then we can then ask, what if imaging were essentially free? What does that open up? So before we can answer that question, we need to ask the question of what makes EM desirable for this task, because it's one of the highest resolution imaging tools available. Most people know that EMs are high resolution, but along with strengths, there's numerous drawbacks. As for strengths, it's known as the gold standard of imaging. It has really high native resolution of about 1 nanometer, strong scattering and contrast, which is about 1,000 times that of light. And one of its key advantages is membrane and surface sensitivity. That is an advantage over light microscopy, which can get point resolution down to 1 nanometer or so with super resolution techniques. But this kind of gets you everything. And so if you want to know where the proteins are, if you tag them correctly, you also get where the membranes are, which is really nice. And at the same time, multiple useful signals are emitted. After its invention, the machines essentially stayed the same for 60 years until the 2000s, when they witnessed a renaissance. And this came because of adaptive corrective optics inspired by the Hubble telescope and availability of cheap computation. And the price just kept going up. It went up by about 5 to 10-fold, and due to the vastly improved resolution. So they're quite pricey instruments. So due to its resolution and membrane and surface sensitivity, it remains a gold standard. And despite it being hugely innovative, hugely impactful in terms of capability, it is actually declining in utilization because of its high cost. This is especially true in clinical medicine. That's a really good case in point where money and time and information may mean life or death. And so in clinical medicine, where it was previously widely used, it's only really used now in 2 main areas. One is in ciliary dyskinesia, which is a mucous membrane disease. It affects the cilia that sweep the mucus around in children, and it's a difficult disease to treat, and also renal disease, where the resolution needs to be very high. And the reason why it's less used is because tool and labor costs are high, and especially since the dominant way EMs are used, which is manual and slow, and high variability and subjectiveness in biological systems has impacted interpretability of results. Like sometimes 2 people will come to different conclusions. And other techniques were started. We're starting to get faster and less expensive and more user-friendly, like genomics. So the direct imaging is a lot less used now, despite being very, very popular in the 60s and 70s. For example, Ebola was imaged for the first time with electron microscopy, and also all the mugshots that you see of the pathogens that are coming out that the CDC looks at. Those are actually also electron microscope images. And so the slowness and the cost have great impacts, especially if you keep the human operator in the loop. And so multi-order of magnitude improvements in speed are very clearly needed. So to solve the problem of mapping in the area of neuroscience, we need speed. And it turns out that I happened to be working on this problem already at the time. Around 2010, I was looking for something new, and having just worked with a team that built the highest performance scanning transmission electron microscope in the world, I eventually realized that most of the interesting problems were in biology, where things weren't so tidy like in material science, where it's possible to get representative sampling of the system with just one image, as I mentioned earlier. The variability of nature is extremely vast, and that was a really interesting challenge to me. So a Silicon Valley startup called Halcyon Molecular was just booting up, and it was trying to execute Feynman's exact idea of looking directly at the DNA basis. And the task was to supercharge the electron microscope to operate much faster in order to accurately sequence enormous lengths of DNA. Hertz Fellow Adam Marblestone was involved at the time at Harvard with the Church Group, and it was a wild 2-year ride where we actually ended up sequencing the bases of DNA, 130 kilobases in length using the TEM. And almost $30 million was spent on this huge multidisciplinary project. And there were parallel efforts in stretching and linearizing DNA, substrate technology, automation, and bioinformatics. And at the end of the day, it turns out Feynman was wrong. It turns out 100x better was not enough. We needed to reach about 10 to the 8th in speed over traditional pipelines. So that's about 100 million. And we had reached about 10,000 to 100,000, which wasn't enough. And in the end, the cost of sequencing was decreasing so fast, about 5 times faster than Moore's law, that it was no longer making economic sense to continue the project for that market, and it was shut down. But it did turn out that the 10,000x speed improvement was enough for neuroscience. And at the time, investment in neuroscience was starting to ramp up. Obama had an initiative that invested 300 million into the area, and Paul Allen had just put 100 million and concluded a 10-year project where they were doing calcium imaging and created a beautiful map that many PhDs used for their theses, and researchers still use it to this day. And at the time, Allen put another $350 million into the institute. So we thought we could help. And so after a reorg and through various channels, including the generous support of Hertz Fellow Ed Boyden, we refactored the technology and got into the brain game. So now we're in the business of supercharging electron microscopy for brain scanning. So the ultra-high throughput technology was a natural fit for the project, and we customized the system for neuroscience, focusing on just a few key areas. So working with the Allen Brain and a couple universities, the pipeline was able to deliver the necessary automation and pipelining, scalable parallelization, and computation to make imaging and reconstructing the large 3D neural circuits highly efficient. So there were several volumes that our instruments helped deliver for them. And this is quite a big project. And we received about $18 million from the IARPA Microns project to perform this. And there were many different collaborators. And in the end, as part of executing the project, the pipeline achieved a full millimeter cubed. And that was just released a couple months ago. And what's really innovative about that is that it covers all 6 layers of the optical cortex. And this was acquired using a fleet of 6 ultra-high throughput microscope nodes. So you can see here, this is a picture of the nodes. And there you can see there's 6 of them. They're kind of old school TEMs. And each one has been modified for speed with a VOXA grid stage reel system. And we have automation through the blade and imaging pipeline technologies that we've retrofitted to the systems. So I'm going to switch gears here, and I will share this link with people through the chat. And you guys are welcome to try this out. So I'm going to change my screen. So I'll take you on a little tour of navigating this data set. It's really fun. Okay, here we go. Okay, so here's a portal, and there's some really neat data on the data set and how it was acquired. And there's a couple of papers that were released at the same time related to the data set. But this software is visualizing the data set. It's about 2.2 petabytes in size. And like I said, it scans 1 cubic millimeter. And it is being served from a Google application called NeuroGlancer that was made specifically for aggregating this type of data and reconstructing and serving it and showing it. So on the left-hand side is a 2-dimensional scanned image. And if you do control click, you can zoom out and see the scope of the data. Hopefully this is easy to see. And you can see the edge here. And we can just keep moving zooming in and looking closely at it, you can navigate around, and on the right is the 3D representation of it. So on the left, if you click, you can see another neuron that gets activated. That's the cell body. And we can zoom in and look at how they're sort of in intimate contact with each other. And you can also see how the dendrites go out, as well as the spikes that are reaching out for connection with other neurons. One of the really neat things about this is that it's not just useful for learning about the interconnections between the cells, but it also shows all the detail about the intercellular and intracellular bodies. And depending on the stain protocols, there's quite a lot of interesting stuff that's going on. And even at the mesoscale, so for example, if you look at this here, this blue circle, if you click on that, you can see the whole volume lights up like a Christmas tree. And this is the vasculature of the system. So you can see the capillaries branching off. And also what's really, really interesting is if you zoom in, you can see these blobs here that show the resource delivery system of microcapillaries in the area. And if you just kind of scroll around here and look at some of these volumes and activate them, you can see that that pair has its own microcapillary network that's supplying it with nutrients and supplies. So there's all sorts of really neat stuff you can do looking through these volumes. To me, you know, as a material science, this is just really exciting because the sky's really the limit to study some of these systems in ways that one has never studied before, because they're just so interconnected and so intricate, and all the detail is there. Okay, so I'll switch back to the presentation. So yeah, I definitely invite everyone to spend more time on that if you'd like. It's really fun to look through and just, you know, challenge your textbook knowledge, and also really easily you can just make little discoveries that that were kind of not expected, and the variability is just really amazing. Okay, so I'll switch gears here and talk a little more about the technology that enabled this. The imaging system is based on a conveyor belt sample delivery system, and the speed of the system when it acquired this particular data set that I just shared was about 100 megapixels a second per node, and we had 6 of those nodes operating in tandem. So here's just a quick video of this operating in 2 dimensions. And so each of these acquisitions is happening in tandem on each of the systems in parallel, and it's acquiring, in this particular case, around 350 megapixels a second. So this is a later version that can go quite a bit faster. And we've been able to hit about 400 megapixels per second in terms of acquisition rate, and that's pretty darn fast, especially for the price of the system in comparison with other types of competing systems based on scanning electron microscopy. All right, and I've already shared this, but this is the navigation of the neuroglancer interface to inspect interconnections between cells. the ability to get speed comes primarily from pipelining, and workflows are really, really, really important. And every step of the way requires an impedance match between the previous step, such that nothing falls behind and slows down the system. So what the process involves is to take a section of brain and section it, and then embed it in and stain it, and then it gets sectioned on an ultramicrotome that picks it up and deposits it onto the conveyor belt system. And it's just like a reel-to-reel tape that used to play music or movies in the old days. And that gets moved over to an analysis pipeline that does the capture. And that happens as in the previous video, and that data gets dumped into a large data store at a rate of several hundred megapixels per second. The post-processing system will do montaging, and then a reconstruction system will put it back together again. And so the idea is at the end, you get a beautiful model. So it's basically pipeline conversion of atoms into bits. And in the end, what's really nice about the use of the TEM is that this is a movie that you can play back again. So if you missed anything, you could go back and image it at higher resolution, or re-image it in different ways. And there's even potential for staining it a different way to do multiplexing. This is what the tiling ends up looking like, and these montages get created. The resolution is about 4 nanometers, and the areas that we acquire currently can exceed 2.5 millimeters squared, and that helps us get volumes pretty quickly. The scale is through parallelization, which is we use low-cost standard TEM platforms, and usually these are remanufactured ones, so the cost is actually fairly low, and it scales with customer needs. So we call it the blade because it's like a blade computing system, and in a blade computing system, each blade node is something that you add with capacity as you need it. And so the way that our customers have been acquiring the systems is usually they'll start with 1 or 2, and then they'll boot up their system, and then start buying more, and then end up with fleets. It's very similar to supercomputing types of models. So now let's talk about going towards whole brain volumes. And so this particular volume was millimeter cubed, and the amount of time it took to image was about 6 months, imaging on 4 of the 6 nodes. The total number of sections was 26,500. There were 125 million images, and like I mentioned, it was 2.2 petabytes. It's not without loss, so we did have some section loss, but it turns out it's quite similar to gene sequencing in that if you have neighboring tissue, you can error correct. And that was a boon to us because everybody thought that you couldn't lose anything. And obviously it's not favorable, but if you lose one section in a row, it actually doesn't kill you in terms of reconstruction. And also I should mention again that re-imaging was actually really, really beneficial. And that's one of the advantages over other methods such as scanning electron microscope based ones, which are generally destructive. In transmission microscopy on conveyor belts, you can re-image. And so there was a re-imaging rate of about 10%, and that's not too bad in terms of total imaging time. So from here, we can say, what does it take to go to the next level in terms of whole brains? So earlier, I think it was released last year, there was a really neat paper called The Mind of a Mouse, and it was released by lots of powerhouses in the field. And it kind of started a conversation of what does it take to actually scan a whole mouse brain? And the federal government got interested in this. I'm not sure if they still have the appetite for it, but it is really exciting to think about the idea of mapping a whole mouse brain. And so if we look at the relative sizes of neural volumes, there's quite a bunch of different ranges of the model systems. So, a nematode nervous system is quite small. It's 300 neurons. And the fruit fly brain, which is quite celebrated right now, there's been a couple of great volumes that came out. One was a hemi brain and one was a full brain. That's about 100K neurons, and the size is quite small. And zebrafish is also pretty small, about 1.7 millimeters squared. But when you start looking at the mouse brain, it's pretty big. It's that the adult mouse brain is about 500 millimeters squared, so it's about 500 times bigger than what we did. And now, what does that look like in terms of an actual project to scan the whole mouse brain? And if you run the numbers, we have newer systems that can go quite fast. So they're going about 300, 400 megapixels a second. So it's faster than the other system. And pretty quickly, you can get improvements in speed that kind of multiply with each other to get you factors of 10. So it is quite feasible to do in, let's say, a year, a whole mouse brain with 100 systems. And that's kind of within the scope of a government project. It's not that big. The human brain is still way, way out there. That's really quite large. But some of these smaller mammalian brains are actually within reach. And if you just look at the pace of technology over the past few years, 2 years for maybe it was 2 years ago, I think, the hemi brain of the fruit fly was acquired. And that was a 20 terabyte data set, and that took 2 years to acquire. The next one was 16 months, and that was a 25 terabyte data set. And today on our systems, our customers are getting upwards of 40 terabytes a day. So it's possible for them to actually acquire entire fruit fly brains within less than a week, quite easily, as technology advances.
Hey, Chris, I
Yep.
hate to, this is fascinating. We do have several really good questions, and
Okay.
we're down to about 15 minutes unless people are willing to stay over. I don't even know if you have time to stay a little extra, but I would love to segue to that, sooner than later if possible.
Oh yeah, sure.
Okay.
Well, I guess I'll just move pretty quickly through the next few slides, and I only have a few more left. And the point is just to show you what the hardware looks like. These are different types of retrofits. The first one is what we call the grid stage sprite. This was a boot up system that we used to do 4,000 tissue sections. It does 16 samples a load, and it's suitable for less than 5K sample size data sets. This tends to be attended loadings, but it's really nice for combinatorial work. Here's the conveyor belt system, and this is suitable for larger sample data sets, maybe up to 100,000 samples, and it has up to 5,500 samples per load. And the speed that we were able to achieve is about a million times the normal conventional workflow speed. We have some boosts in speed as well from just messing around with the optical systems. We can make the thing go quite a lot faster by mixing and matching the piezo stage and the electron-optical systems. So here's just an example of how fast we can scan. And this is the conveyor belt system in action. So a fundamental change in paradigm here is just offered by the fact that sample exchange is effectively effortless. It happens within seconds. So that downtime is eliminated almost entirely. Eventually, it kind of becomes the dominant duty cycle limiter. Okay, so let's just talk very quickly about future application spaces. These are places that we're really interested in going at, as a business. Obviously, larger volumes with more microscopes is really exciting, going to larger brains, but there's also a lot of other vectors that we could explore. For example, many small volumes. If it takes less than a week to do a small volume like the fruit fly brain, then biopsies could be done, 50 to 100 a year on one node. That's pretty interesting. There's also other areas in material science, such as environmental monitoring, like pollution screening for like asbestos. There's also manufacturing QC/QA. We already mentioned drug development pipelines, but also in the research world, basic science, combinatorial chemistry, as well as material science. And that is really exciting to be able to look at nanoparticles at that kind of speed. So what's possible here is time evolutionary studies, or temperature, environment type of changes over different sample series. And that kind of stuff wasn't available before because sample exchange was really hard to do. So now we can do that. So that concludes my my talk. And so let us know if we can help you. So Vox's mission, as I mentioned, is to increase access to advanced instruments and measurement data to catalyze new technologies and improve the human experience. So also we're hiring. So if you're interested in working with us on this problem space, we would love to hear from you. So with that, thank you very much for listening, and I'm happy to do some Q&A.
Great, thank you so much, Chris. I want to dive right into the questions.
Okay.
Joe Sherry, you had a question first. Are you still here, and can you answer your question or pose your question to Chris, please?
Yeah, sure. So you actually touched on this a little bit after I asked the question, but I'd be curious to hear more details. So when you're tracing very thin processes over long distances in these large-scale samples, I was wondering what your tolerance is for artifacts that interrupt those processes and affect the circuit reconstruction, and then how that compares to like the actual imaging and stitching artifact rate.
That's a great question. So I'm afraid I'm not super qualified to answer that, because our specialty is mainly in the 2 dimensions, and we're not actually doing the 3-dimensional reconstructions. That requires huge computational resources. So that's actually being done by a company called Zeta out of Princeton. And there is this thing that's known in the field. It's called the tyranny of scale. There is one fellow from the Denk group in Germany who talks about this a lot. And it's basically saying that like you can trace with some probability and some reliability up to a certain extent, but it falls off pretty quickly. So there's certain types of neurons that they call them cortical loops, which are just dastardly super difficult. And there's already this issue of how do you section a let's say a whole mouse brain. We haven't, as an industry, solved the problem of how you thin section or how you section larger volumes. We call this subsectioning, that the thin sections are cut from. So there's this issue still remains. And so there is some limit to how, like probabilistically, how many sections we can go before we lose track of things. So far, we've seen that it's probably larger than 1 millimeter, and probably by a lot. But when you're going into the centimeter range, the fall off, I think, is pretty quick based on calculations. So that would be a really interesting machine vision problem, because the data is there. It's just how do you deal with distortions, and how do you deal with problems with the physical processes tying into the software processes later to try and stitch things.
Mike Worth, your question's next, if you're still here.
Yes, I am, and this has more to do with results of your scans rather than your technology for measuring. And that is, given the hierarchical nature of connections, are there enough of them? And are they self-similar enough that the mathematics of fractals becomes a useful modeling technique for the overall structure?
That's a really good question. So I'm an engineer, not a mathematician, and also not a neuroscientist. So it's difficult for me to answer that directly. There are people in our groups, in the collaborating groups who are working on these things. The resource base is a little bit tight in that most of the approaches are naive. And, so it's sort of trying to brute force things. And there's always this emphasis on, perfect pixel or perfect voxel integrity. And I have a sense that there is a lot in terms of a priori information that hasn't been put in to these sorts of reconstructions. And I think that nobody in the field is really working on it. when I was working in diffraction, this was old hat. We did this all day long because you had to use a priori information in order to reconstruct the phases in order to create structures. And so in protein crystallography and that sort of thing, that's done all the time, right? Using mathematical support constraints to help you narrow things down. So a lot of people are trying machine learning approaches, but as far as I know, nobody's using fractals to try it. But I think that there's this whole effort of getting the data, and then there's this question of who's going to go in and study the data in different ways, look at it in different ways to start pulling out these sort of hierarchies. And I think it's early days yet. So I'm not sure if I answered your question, but hopefully that gave you some context.
No, thanks a lot. To be really esoteric, I'm thinking back, I'm an old guy. I'm thinking back to the 70s and 80s, Martin Gardner's column in Scientific American. And the question is, what's the relationship between music and fractals and the brain? So there you go. There's a wild thought.
Yeah, I mean, there's so much hierarchy that I would not be surprised if somebody found a system like that. I mean, we found a compressed sensing circuit, like just textbook, right? And there's just, it's going to solve evolutionarily these problems in just myriad ways. And there are some things that we can't even see through structure, right? There's chemical sensing as well that is a little bit hidden. So I wouldn't be surprised if that comes out of this at some point.
So, by the way, wonderful technology.
Oh, thank you.
We do have several other questions, and I just want to let everyone know we can stay over if Chris is available to do that. So we're going to keep going. David Cohen, you're next if you're still here.
I'm definitely still here. Thank you very much. That was fascinating in all respects. So from a both scientific and personal family interest, my question is, are your technologies potentially applicable to diagnosing and in particular evaluating treatments for multiple sclerosis and other demyelination diseases?
I would suspect yes, because stains can be selective to myelination.
Right.
And there is interest in this. This is the biggest data set, and nobody really is doing small data sets yet because everybody's been trying to get the really, really big one, right? And so there's this question of, are you going to be first? Are you going to be best? Or are you going to be most? That's the question in the research world in terms of having the crown, right? And so I am, as a business person, but as well as somebody who's tries to sort of think outside the box, I think looking at small volumes across many, many samples is the most interesting place for me. One of the big problems of this is an ex vivo technology. And that means everything's dead, and you have to sort of make sure that it's a pretty decent representation of what it was when it was alive. And then second, if you are a sufferer of MS or related disease, you probably don't want brain biopsies. And so this is the kind of study that has to be done on cadavers. And that's actually a really interesting area because most people who are doing work in humans, for example, have to work with cadavers. And at the moment, there isn't a huge availability of human tissue for that kind of purpose.
Yeah, if I could just follow up quickly, if I leave out the diagnosis part, but evaluating treatments, I mean, most of the treatments, potential treatments for MS are first evaluated in mouse models.
Yep.
So that is then avoids the problem of waiting until the human patient is beyond benefit.
That's right. That's right. So I think this is pretty new as yet, and I would love for more people to be interested and engaged in this. I got into it because I'm very interested in disease and helping accelerate therapies and understanding disease. Most of the people in the field are interested in structure function. obviously you need that because they're concerned with ground truth. But I think like the end game, at least for me, is can we solve some let's say degenerative diseases that affect so many people, and it seems to me that there's a lot of low-hanging fruit there in any sort of model, like mouse models or human models as well.
Chris, quick question.
Hi, David.
Hi, how are you?
I'm doing great.
The great talk. Thanks a lot.
Thanks for listening.
My question is anybody looking at applying this your technology to worms and flies, because that's a huge area of research in Cinorhabditis and Drosophila to try to understand the, I mean, being able to look at the circuitry is a piece that's often missing in the research, and it would be just fabulous if I think your technology would be really cool. I'm not sure how you automate the samples with them, but there's an awful lot of people working on that. Are you aware of any activity there, Chris?
Well, the worm model and the fly model, which I mentioned in the talk, are very, very popular. And they're popular because they're small and they're also fairly interesting. As far as models go, the inputs and outputs are very interesting to measure, and they're with context of behavior and all that. I think you hit on one major point, which is sample preparation is a major part of this, and our part of it has been in the imaging automation and the sample delivery part. But what we do is we receive the samples from the user, and they have to understand how to manage the tissue, and embed it and stain it properly before it comes to us. So I think that's still, a frontier in terms of the technology's usefulness and adoption. What we're trying to do is make the imaging very, very low cost in relation to everything else, so that adoption could be accelerated. And low cost is subjective, of course, because these are still electron microscopes, but they just go a lot faster. So you get a lot more for your dollar when it's sitting in your lab and you're paying the electricity bill on it. But to answer your question, I do expect, more results from flies and worms probably. There are people who specialize in that. But I think the cool thing is that now some of the larger brain circuits are within reach. And so there is probably going to be, my guess is that there's going to be more movement towards larger volumes for some time.
Great, thanks.
Frank Wertherween, I'm not sure if I'm saying your name right. Sorry about that. Are you still here?
He's
Frank?
left, but I was instructed to ask the question on his behalf.
Great.
So his question was, can you expand on how this enabled re-imaging? Naively, this requires running the conveyor belt backwards, finding a previous 2D slice, and then re-imaging it. So does this imply that you can store sort of the process? And for one sort of millimeter cubed or 125 million slices, do you have a single conveyor belt or a single process that long?
So to clarify, it's not 125 million slices, it's 125 million images, of which each slice integrates 10,000 of in this particular setup. So it's actually 27,000 or 27,500 tissue sections. So it's a lot more manageable than 125 million. And so they pick those up and store them onto, I think, 5 sets of reels of 5,500 each. And those get, stored like old movie film canisters, at Hollywood. They store them in sort of a nitrogen environment, and they're all indexed, and there's information that's stored in software that's related to each indexed sample. So we offer the ability for random access, so you can go back and find a particular section as long as the tape is ID'd, and then we can go forward and backward and find that thing within some fairly short amount of time and re-image it based on a program that was stored before. There is some change to the tissue because of the energy of the electrons, and one has to sort of take that into account. So each time you re-image, the tissue shrinks a little bit more. There's some cross-linking that happens to the polymers, but that can be addressed in algorithms later on.
Thank you.
Andrew Teal, are you still here?
Yes, I am. Thanks. I wondered about sample preparation. I think it's already been discussed, but wondered if you could embed markers, kind of like survey markers or benchmarks, into the tissue before sectioning and scanning it, so that you could use those markers to help reconstruct the images.
Yeah, that's a great question. It is done, but it's harder to do than one would expect. And we've thought about some solutions for this, like technology solutions, and we just haven't done enough volumes to try something super different from the normal embedding operation. What's done sometimes is people will take an eyelash and embed it, and that becomes a fiducial. What the Allen Brain does is they bevel the corners of the block, and then you get kind of a pointy shape that you can use for fiducials. One of the big issues is that when you're slicing, we're slicing very thin, so it's like 50 nanometers is what they slice at. It's tremendously thin, and we are actually working on ways to make that relaxed more. And so the distortions happen in lots of different ways, because this is quite a high energy operation to slice with a diamond knife at 50 nanometers thickness. Basically a membrane that will just like burst, if you look at it the wrong way. So there's quite a lot of artifacts that come in, and the fiducials would definitely help a lot. But I kind of think you would need more fiducials for the registration. And in the end, the axonal arbors and that sort of stuff, like the vasculature is incredibly helpful, because that's already kind of built in as a fiducial between layers. So as far as I know, they don't tend to use it for their reconstructions. Nobody's asked for it.
Thank you.
This last call for any other questions. We do have one more that was in the chat box from Soren Emerson. Are you still here?
Yes, I am. Hi, Chris. Great talk. I'm coming at this from the neuroscience background, and I understand you are really coming at it from the engineering background. So I apologize if this is a little bit out of directly your focus. But at the beginning of the talk, you made an analogy between making maps of the geographic world and making maps of the interconnections between neurons. And one key difference between maps of the world and maps of neurons is that the interconnections between neurons change all the time, depending on basically on the experiences of the organism. So when you're making these maps, I was wondering if you'd thought about different ways that you can effectively make an average of what the interconnections look like to maybe account for some of the changes in structure induced by experience.
Well, I did mention that this is ex vivo, and the fact that the tissue is dead and it has some sort of evolution between the time it was sacrificed and the time it was embedded, there's always this question of, what's the integrity of the connections? You might be coming from an in vivo type of background, and the dynamicity of the systems is just incredible, right? It's just constantly changing on the order of seconds, right? And so I think for measuring the connections, what we do is actually probably a boon to the system, in terms of understanding what's going on because it's frozen. So the question becomes, is there a change between when the critter was sacrificed and when it was embedded? And I'm not sure if we have ground truth on that quite yet. But what they are seeing, though, from the results is they are able to reconstruct circuits. And you know, there is still, you know, there are still issues in terms of making sure that we get the detailed interconnections, because there are things like gap junctions and, you know, different flavors of connections that aren't super easy to see even at this resolution. And it's, as a community, they've all kind of fixated on this number of 4 nanometers, but it turns out that in some situations you need 2 nanometers or 1 nanometer. And there's a question of, are you actually catching all of them? And you know, they caught like 500 million or something with the automated processes for machine vision, but I'm sure we're missing a lot of them. And there's still a lot of room for the technology to improve to get that. And you know, the scary thing is that if you go down by a factor of 2 in resolution, or go up by a factor of 2 in resolution. So let's say you go from 8 nanometers to 4 nanometers, you have to spend almost 10 times as long imaging to get all that data. And so there's opportunities for speeding up that we haven't totally explored yet. But hopefully to answer your question, there isn't going to be an averaging of the connectome, so to speak, as far as we know, because we think that what happens is that it's a snapshot at the time of sacrifice and then it's frozen.
Well, thank you so much.
Of course, thanks for your question.
That is all the questions we have so far. So Chris, thank you. Fascinating. There are a lot of really nice comments in the chat just thanking you and how fascinating it was. And we will have the recording available and on our website, I'll say in a week's time.
Okay.
So check back for that. And we do have this next event happening October 13th with Leonid Krugliak. So hopefully we'll see everyone there. And if you did not receive the notification or the announcement on the application, please let us know and we can get that to you so you can share with others. Robbie, did you want to sign off, say something?
Just a hearty thank you, Chris, on behalf of all of the attendees. I know some folks had to jump off and move on to other things. And also a big thank you from the Hertz community, making this talk available later on through recording for others. I know is going to be really important. I've gotten personally a number of inquiries saying that they couldn't attend and they were hoping that you would allow us to record. So again, thank you, but also congratulations on this incredibly important work. You and your colleagues are making terrific progress, and we appreciate you sharing that with us. We wish you well.
Thank you so much.
Thank you, and best wishes to everyone.
Bye-bye, everybody.
Bye-bye.